Welcome to Developmental Biology. Today, we're going to explore one of the most universal and fascinating mysteries of life: why and how we age. We will uncover the biological clocks ticking inside our cells and the mathematical laws that govern our lifespan. By the end of this lecture, you will understand the fundamental theories that explain the inevitable journey from peak vitality to gradual decline. Let us start by imagining a brand new smartphone. Out of the box, it runs perfectly, the battery lasts all day, and everything is fast. But as the years pass, the battery degrades, the software slows down, and it eventually stops working. This everyday experience is a perfect analogy for the biological process of aging. Just like that phone, our bodies accumulate microscopic damage over time. Our internal repair mechanisms, which once kept us in peak condition, gradually lose their efficiency. This inevitable journey from peak vitality to gradual decline is exactly what developmental biologists study when they look at aging. Now that we have a general picture, let us define this formally. In biology, aging is known as senescence. It is the progressive, time-dependent deterioration of physiological functions necessary for survival. It is not just a single disease, but a complex systemic decline affecting virtually every tissue and organ in your body. As an organism grows older, its cells lose their ability to divide, and its immune system weakens. This leads to a progressive loss of homeostasis, which is the body's ability to maintain a stable internal environment. Without this balance, the organism becomes increasingly vulnerable to environmental stress, diseases, and ultimately, death. Understanding this systemic decline brings us to an important question. Why do we study aging so intensely? If we do not understand how our cells age, we cannot effectively treat major age-related diseases like Alzheimer's, cancer, or heart disease. But scientists are not just trying to extend the human lifespan. More importantly, they aim to increase the health span. Health span is the period of life spent in good health, free from chronic diseases. Furthermore, studying aging helps us understand the evolutionary trade-offs organisms make between reproducing early and maintaining their bodies for long-term survival. We want to treat the fundamental biological clock, not just the symptoms. This idea of a fundamental biological clock leads us to our first major theory at the microscopic level: the cellular theory of aging. Every time a cell divides to repair or grow tissue, its DNA must be perfectly copied. However, the enzymes responsible for copying DNA have a flaw. They cannot reach the very end of the chromosome. To protect vital genetic information from being lost, chromosomes have protective caps at their ends, called telomeres. You can think of telomeres like the plastic tips at the ends of shoelaces. They stop the DNA strand from unraveling and fraying during the copying process. Building on the shoelace analogy, let us look at what happens over time. Because the copying enzymes fall short, a tiny piece of telomere DNA is permanently lost during every single cell division. As we age, these protective plastic tips get shorter and shorter. Eventually, they are completely gone. When the telomere reaches a critical minimum threshold, it triggers a state called cellular senescence. At this point, the cell permanently stops dividing. This is a protective measure to prevent the cell from copying damaged DNA. But it also means our tissues can no longer regenerate, which is a primary driver of aging. Now that we understand the mechanism of telomere loss, Let us look at its mathematical representation. This formula calculates the length of a telomere after a certain number of cell divisions. The variable L of n represents the final length of the telomere. L0 stands for the initial, starting length before any divisions occur. The variable n is the total number of times the cell has divided. Finally, delta L represents the constant amount of DNA lost during each single division. Conceptually, the current length is just the starting length minus the total DNA lost. As the number of divisions increases, the final length decreases until it hits that critical stopping point. Before we move on, let us see this formula in action with a concrete example involving human fibroblasts. We are given an initial telomere length of 10,000 base pairs. The cell loses exactly 100 base pairs per division, and it will reach senescence at a critical length of 4,000 base pairs. We need to find n, the maximum number of divisions, known as the Hayflick limit. We substitute our known values into the equation. Next, we rearrange the terms to isolate the total loss, which is the starting length minus the final length, giving us 6,000. Dividing by the loss per division, we find that n equals 60. This means the cell can divide exactly 60 times before aging takes over. We've just seen how telomeres explain aging at the microscopic level of a single cell, but we also need to understand how aging affects entire populations. In the 19th century, a mathematician named Benjamin Gompertz observed a fascinating pattern. He noticed that once humans reach adulthood, the probability of dying does not stay constant. Instead, it increases exponentially as we get older. This observation became the Gompertz law of mortality. It shows that aging is a systemic acceleration of vulnerability. The body's ability to repair itself declines at a compounding rate, very much like compound interest on a financial debt. This compounding vulnerability is perfectly captured by the Gompertz law of mortality equation. This formula models how the risk of death multiplies over time. The variable m of t represents the mortality rate at a specific age, t. The symbol m0 is the baseline mortality rate at the onset of adulthood, representing non-aging risks like accidents. The constant e is the base of the natural logarithm, driving the continuous exponential growth. Finally, alpha is the Gompertz coefficient, which is the actual rate of biological aging. If the aging rate alpha increases, the mortality rate shoots up much faster, meaning the population dies at a significantly quicker pace. Now that we have our exponential formula, let us apply it to a hypothetical population of laboratory mice. We are given a baseline mortality of 0.01 deaths per year, an aging rate of 0.5, and a time of 4 years. We want to find the new mortality rate. We plug these values into our equation. First, we multiply the aging rate by the time to get an exponent of 2. Evaluating e squared gives us approximately 7.389. Multiplying this by the baseline mortality yields a final rate of 0.07389. Physically, this means the mice's risk of dying jumped from 1% to over 7% in just 4 years due to biological wear and tear. Having calculated both phenomena, let us look at them visually. On the screen, you see 2 graphs that summarize our 2 main theories. On the left, the telomere shortening graph shows a straight line sloping downwards. As the number of cell divisions on the horizontal axis increases, the telomere length on the vertical axis steadily drops until it hits the dashed line, which is the Hayflick limit, where senescence begins.
On the right, the Gompertz law graph shows the exact opposite shape. As age increases on the horizontal axis, the mortality rate on the vertical axis curves upwards exponentially. Together, these diagrams show how linear cellular decline translates into an exponential increase in organism vulnerability.
We have covered a lot of ground today, so let us summarize the most important ideas. First, aging is a multi-layered phenomenon happening at both the micro and macro levels. At the microscopic level, telomere shortening acts as a built-in biological clock, limiting tissue repair through the Hayflick limit. At the macroscopic level, the Gompertz law of mortality proves that this cellular decline translates into an exponentially increasing risk of death for the entire organism. By combining cellular mechanics with population mathematics, we gain a comprehensive understanding of senescence. You now have the foundational knowledge of how and why organisms age, paving the way for future breakthroughs in developmental biology.